BioSpectrum India. Before we start the webinar, there are a few house rules which I would like to take you through. All participants will be at listen-only mode. If you have any questions, please wait for our Q&A slide to pop up. You can type your Q&A into chat panel. This webinar is also available on demand on biospectrumindia.com. In this webinar, you will hear from Dr. B.M. Rao, Head of QA for Emerging Markets, Analytical Science and Technology, and Corporate Quality Control at Dr. Reddy Laboratories. And also, we have Dr. Anup Kumar, Business Manager, Sciex India. Let me pass floor to Dr. B.M. Rao to start the webinar. Hi, Dr. B.M. Rao.
Good morning, everyone. First of all, my apologies for some issues on the network. So I'll just start my presentation. I would like to thank Sciex for giving me this opportunity to lead my sharing on this nitrosamine impurities. My experience on nitrogen impurities in API and pharmaceutical products and how to deal with them. Please go to the next slide. Yeah, this is a disclaimer. Go to the next slide, please. So in this presentation, I will talk very briefly on the guidance document. Sorry, I am getting a lot of echo.
Sir, go ahead. We can hear you. No problem.
Yeah. In this presentation, I am going to cover guidance documents, list of nitrosamines, what are there and what are the potential sources, risk assessment, recommended methods, what are the analytical methods, and what are the practical challenges we see, and what is the suggested approach, robust approach. Are you able to hear me, Anu? Hello? Okay. So these are some of the guidance documents published by various regulatory agencies. The first one is the European Medicine Agency has published a document. Then Health Canada published a document. FDA has published, of course, testing methods as well. And just I want to emphasize one thing. Couple of days back, that is precisely on 24th April, even USP announced that they are going to publish a general chapter on nitrosamines. And they are asking inputs from the industry. The deadline for the input, getting inputs from the industry is 22nd May 2020. So this is the latest one. Please go to the next slide. So this is the introduction wherein, so maybe all of you are aware that the nitrosamines basically molecule which contains the nitrogen functional groups bonded to deprotonated amine. And since it is a carcinogen, it is really a concern. And there are so many reports in the literature saying that the presence of nitrosamines are there also in the food and drinking water. But however, in the medicines, it is considered to be unacceptable. So if you can see the recent potential issues in 2018, we all know that there are a lot of concerns came on this presence of nitrosamines in sartans. Then also later it was ranitidine, a lot of issues, and many companies have initiated the voluntary recalls, including Dr. Reddy. We have also initiated the recall, and we have recalled a lot of batches of ranitidine from the market. And later regulators have now have a stringent manufacturing requirement for the medicines. Next slide, please. Yeah, this is very important slide. What is the root cause for the presence of these nitrosamines? So the first one is use of sodium nitrate in the manufacturing process, and then whether somebody is using the raw materials in the API manufacturing process. For example, it can come from the solvents, reagents, and catalysts, and use of recovered solvents, which is very critical, which can contribute to the presence of these nitrosamines. Not only recovered solvents, it can include the reagents and catalysts. Then we have there can be use of contaminated starting materials and intermediates. And there could be sometimes the cross contaminations in the manufacturing facility, which can lead to the, especially when different processes are running on the same line, there could be a probability of the cross contamination. Then there can be also possibility of the formation of nitrosamines. Then the degradation of process of starting materials, intermediates, and finished products also can lead to the formation of nitrosamines. Last but not least, it was also noted that the primary packing materials, which contains nitrocellulosic material, which can react with the amines in the printing primer, can also form nitrosamines. These are the probable root causes for the presence of nitrosamines in the API or drug product. Please go to the next slide. So this is the structure of different nitrosamines. I think most of you are aware that this list has been published, and item 8, 9, 10, NMOR, NP, IP, and NPYR, they are published in the USP. Basically, these three nitrosamines can potentially come from the packaging materials, as I mentioned in the earlier slide. Next slide. So basically, the origin and chemical structure of nitrosamines in sartans. Basically, this to produce nitrosamine, the presence of second amine and nitrate reaction under acidic pH is required, and it is suggested to perform a risk assessment based on the need during the manufacturing process. And after risk assessment, we also do a lot of testing, which I'll cover in the next slides. But here in this slide, interestingly, you can see in the small table where what are the different amines can contribute to corresponding nitrosamine impurities. For example, dimethylamine presence can potentially give NDMA, that is N-nitroso dimethylamine. This impurity can come from due to the presence of dimethylamine in presence of nitrous acid. Can you please go to the next slide? This slide talks about the EDQM, European Authority, has given a guidance how we can efficiently avoid the nitrosamines in human medicines. So they proposed a three-step process. Step one is basically we have to conduct a risk evaluation. Industry has suggested to conduct a risk evaluation to identify the products of where there is a possibility of the formation of nitrosamines, or is there any possibility of the formation of nitrosamines because of the cross contamination, because sometimes in the API facilities a lot of multi products will be manufactured in one line. So initially they gave a deadline of 26th March 2020. Later the date is extended for the risk evaluation till 1st October 2020. Then the second step is after identifying the risk evaluation, it is also suggested to do the confirmatory testing of the products where there could be a probable presence of nitrosamines. And the third step is after understanding the from the confirmatory test, if there is a potential formation and analytical test also confirms there is a nitrosamines presence, the step 3 is industry is suggested to change the manufacturing process to make sure that the level of nitrosamines are controlled well within the acceptable limits. So these are the three-step approach EDQM has suggested. Then this slide is very, very important. So basically, I am talking about risk assessment and control. There are two-step process. One is step 1, where we do as a part of risk assessment, we do the risk identification, risk analysis, and risk evaluation. And then as a part of risk identification, first of all, we should do various assessment of various input materials for the possibility of nitrosamines. What are the materials, input materials are there, which are being used in the APIs or drug product manufacturing. Complete assessment of all the materials. need to be done. Then, of course, the process assessment will be done if there is any possibility of formation of nitrosamines during the process. Then, as a part of risk analysis, because drug product contains various components. In addition to the active pharmaceutical ingredients, we use a lot of excipients which come from the different manufacturers. We have the packaging components, and we have the water we use in the drug product manufacturing, and we have the manufacturing components, and we have the cleaning agents what we use in the drug product side, and drug product manufacturing facility. A lot of equipments are also shared. So I think a lot of assessment need to be done with respect to each and every component, including API, excipients, packaging materials, water, to make sure that if there is any possibility of the nitrosamines formation, and this risk analysis should be very comprehensive. And sometimes it is a very, very difficult to get the data from the vendors, excipient vendors, but it is very important to insist them get a lot of data. In case if we don't get the data from any of the excipient vendors, it is also recommended to do the analysis of this drug product for the presence of nitrosamines. Please go to the next slide. So this slide talks about the risk evaluation. As a part of risk evaluation, it is suggested to do a thorough failure mode effective analysis. Basically, it is a type of tool with different scores assigned to various risk levels. Basically, we try to understand what is the risk level, whether it is high, medium, or low, based on the evaluation. Then there should be a thorough checklist also, type of a yes and no questionnaire. Where it can be. I have seen recently there is a publication from ISPE where they have published a nice checklist for the nitrosamines. It is a preliminary list. One can do some risk evaluation based on the process or the manufacturing process. It is a yes or no questionnaire. There a lot of questions have been published. You can say yes and no. Based on that, we can do some level of a risk evaluation. Then, as a part of the risk control, once we have the risk evaluation, it is very, very important to have the appropriate control strategy to establish in the drug product. So once we do the risk evaluation, then we arrive at the appropriate control limits in the drug for the drug product. Basically, it is based on the acceptable intake for each drug. The level of dosage will be different. The acceptable intake levels will be different of the drug. So based on that, we'll arrive at proper limits for the suspected nitrosamine, and based on that, we'll do the testing. If you can see the small table which is there in this one, if suppose my acceptable limit of NDMA for example, in one drug product is 1 ppm, if I am getting less than 30%, then there is no confirmatory testing is required. If it is more than 30%, we'll potentially test and understand how the trend is going to be, and if it is more than the specification limit, each and every batch need to be checked to make sure that the level of nitrosamine is well below the limit. So it is suggested that we'll do 3 consecutive commercial batches of the API to confirm the level of nitrosamine in the API. And once we check in the API, if the level is much below 30% of the spec limit, then we need to have any control. But however, please remember that whenever we change the API process or any changes is made, any recycled solvents has been introduced in the process, it is again recommended to do the fresh evaluation, risk evaluation with respect to the nitrosamines formation. And whenever we get the more than 30% of the internal limits, I think it is also the responsibility of the manufacturer to inform to the marketing authorization holder. Please go to the next slide. And many of you know that the analytical controls will be a real challenge in this, because the limits are at a very low level. Like, it is very, very important to develop a rugged and robust method for the quantification at low levels. So limit these impurities as these limit of 2 impurities are very, very low. High sensitive techniques are required, and we need to ensure that the analyte which we are trying to determine is stable in the solution. So, in fact, FDA has already published some methods, as I mentioned. They have published HRMS methods, they published LCMS methods. Also, some GCMS methods have been published. But however, in our company, we have really we really worked on the lot of methods on the triple quad LCMS-MS. We got a very huge success. We could able to go up to excellent sensitivity. We got very good LOQ and ODs about 0.03 ppm and less than 0.01 ppm sometimes, many times. And we also could successfully establish the sensitivity at LOQ and precision and accuracy. We have very successfully established. I think from the analytical standpoint, one thing is the developing the sensitive method is very, very critical. But at the same time, I think method transferability to manufacturing. Once we develop a method in the R&D, I think it is also very, very important to make sure that the method works in the manufacturing quality control labs. So what we have done is, I think we have purchased the same make, same model mission, what we have in our R&D, so that we don't see any challenges in the method transfer. The method transfers are smooth, and we can, it really helped us in releasing the batches. For example, in one of our manufacturing facility, we are making a huge ton of one API. So we have done recently 300 batches very successfully, and ensured that the potential nitrosamine which is expected to be present is less than the 30% of the threshold limit, specification limit. Please go to the next slide. Then these are some of the FDA published method. I will not go into all the details, but there are, as I mentioned, there is some GCMS methods, there is some HRMS methods, and there are LCMS-MS methods LCMS methods have been published. But from our side, in fact, in our LCMS-MS triple quad, even one of the good thing what we have done is, even we could able to establish the methods, very generic methods, where the up to 5 potential nitrosamines, we could able to develop a very single method, and we could be successfully validate that method. But of course, we have to establish the accuracy, recovery, LOD, LOQ with respect to the specific API. But definitely, we got good success in developing a single method. Please go to the next slide. So some of this slide talks about some of the challenges in method development. I think what we can see is, I think we need to have as I mentioned, we need to develop a sensitive methods. We need to ensure the suitability of the method for the target determination. And sometimes we have a challenges with respect to the solubility of the analyte with respect to impurities. And we need to separate the interferences of the other impurities. There is also a challenge. Especially when we are doing drug product, we also have a challenge of placebo interferences. And since the detector is mass detector is very sensitive, I think we have to avoid contamination. So sometimes we have the challenges of saturation of the detector. And I think it is also challenging to reproduce the detection and quantification limits. So that's why I'm always suggesting to have the same make model instrument at different sites, so that it is very easy to reproduce the limit of quantification, the limit of detection, instead of having different make of instruments. And some of the practical challenges, I think one important is here I want to highlight is the peak shape. Sometimes we get challenges in the peak shape of the particular nitrosamine. So we could able to get some good success by adjusting the organic aqueous ratio. And most important is, I think one of the challenge we are going to have in future is the EMA, European Medicines Agency, is going to put a limit for all the nitrosamines irrespective of any API. It is less than 0.03 ppm. So this is the level we have to have the specification limit. So our limit of quantification should be less than 0.01 ppm. So it is suggested that to have a very good sensitive LCMS-MS or HRMS, whatever it is, it should be able to get the detect and quantify, especially the quantify less than 0.01 ppm. Please go to the next slide. So here I'm going to talk about the drug product. So basically, when we have done, as I mentioned, that some of the few of the manufacturers of the excipients and other components, they are not able to give the declarations. So it is then we have to do the drug product assessment with respect to the probable nitrosamines. So what we have done is, I think here we get some challenges. So basically, it contains also some excipients. I think it is very, very important to make sure that the recovery of nitrosamine impurities at LOQ level is critical, and it is a challenging. And there is an interference of the excipients with respect to the nitrosamine impurity peaks. So this is one of the challenges we have seen. So here I have put some suggested approaches. I think first we need to do comprehensive assessment of synthetic route of API, and also we should understand the toxicological attributes. Assessment we have to do in accordance with ICHM 7. Then we have to do the risk assessment for all steps. What I have mentioned for the synthetic steps, we have to understand where there is a formation of nitrosamine impurities. We have to see the reagents and manufacturing process equipment where it can potentially form. That is very important to understand. Then I think, as I mentioned, development of robust advanced techniques is very, very important. And sometimes it is also important to verify using some orthogonal techniques. Wherever you have any doubt or some suspicion, it is also suggested to have parallel orthogonal technique to make sure that the potential nitrosamine impurity is well within the limit. Then, of course, I already talked about hyphenated techniques like LCMS-MS and GCMS for the quantification, and we should need to have extended linearity range. And as I mentioned, it is important once we develop the LOQ and future, the 100% of the LOQ target should be 0.01 ppm. And once we establish the LOQ, I think it is important to make sure that pressure and recovery and pressure accuracy at LOQ is established very well. And last but not least, I think validation and transfer of the methods need to be done in according with the internal SOPs. Every company will have their own SOPs, but it need to be done very, very thoroughly. I think this is my last slide. Please go to the next one.
So my agenda is like this. I'll be taking probably 15 minutes to complete this, because most of the things you have already heard from Dr. Rao. So we'll be talking about Sciex product portfolio, nominal mass spec and high res, slight a bit, and then introduction of nitrosamine impurity. I'll be also showing you some of the analytical methods of nitrosamine, what Sciex lab has developed. Some 3 example, we will be showing you Losartan, Losartan and ranitidine, and some of the method which we are not showing, you can talk to our marketing team and other team members to get the more information. And then we will be also talking a little bit about the QTrap functionality for the qualitative and quantitative workflow for nitrosamine. QTrap is one of the hybrid instruments what Sciex has, and it's owning the proprietorship of this technology. So we will be talking about this little bit, and some global reference on LCMS method and global Sciex tech note we will be talking in this particular slide. Next slide, please. So if you see, this is a current 2020 LCMS MS portfolio of Sciex, what Sciex owns. Sciex is working in nominal mass platform, where we have a 2 variant, triple quadrupole variant, and a unique hybrid linear ion trap, Q-trap variant, what we have, starting from 3500 to 6500 plus instrumentation. For nitrosamine impurity, mostly 45 and 5500 plus level of sensitivity is more than sufficient. And all the methods what we have generated, and all the method what here in India, a lot of major pharma companies have generated, has used 45 Q-trap and 55 Q-trap mass spectrometry. You may also see below there is a 3 product line, which is high resolution mass spectrometer QTOF based technology, because some of the method what you are seeing that people has done on HRMS. So we have also high resolution mass spec product line to support the method for nitrosamines, but definitely Q-trap and triple quadrupole is most a unique methodology to run any targeted quantitation. Next slide, please. I'll be talking about 2 machines, as I told you, the 4500 triple and Q-trap and 5500 machine in coming slides. So next slide, please. And I just want to give you a little bit technical overview of 4500. This is a mid-segment sensitive triple quadrupole and Q-trap variant. So in 45, it comes with a 2 variant. One comes with a triple quad, and another one comes with the Q-trap variant. What you are seeing is a very compact design with a very better electronics. It has a very fast polarity switching, a very fast electronic for scanning speed. And the most important part of this instrument is that it is coming with a high pressure Q0 and QJet2 ion guide, which help you to focus more number of ions which is generating in the source, and then it is pushing into first quadrupole. First quadrupole is also equipped with the RF frequency of 940 kilohertz. So therefore the mass range of this machine goes up to 2000. Then you have a curved collision cell and third quadrupole. In Q-trap variant, the third quadrupole can switch into ion trap. Therefore, it can allow you to give you a confidence in terms of doing a lot of characterization experiment in the same run. So the Q-trap technology help you to run qualitative and quantitative assay in a single platform. And next slide, please. We have now new 5500 plus launch in 2019 ASMS. So I will be just showing you one slide giving you a technical overview about 5500 plus. So the 5500 plus is a re-engineered, redesigned, a new advanced system with a lot of changes in hardware. The major hardware changes what you are seeing in 5500 plus compared to its previous version of 5500 is that it has a very fast polarity switching of 5 millisecond. It has a dual QJet ion guide, the ion guide which we are using in our super hot and super sensitive mass spec, which is 6500. So same dual QJet guide we have introduced in 5500 plus, and more important thing is to improve the linear dynamic range. We have high energy diode detector technology introduced in this machine, and this machine 5500 plus again comes in a 2 variant of triple quadrupole and Q-trap ready variant. And what you are seeing that it is controlled by analyst software, and data can be processed by Sciex OS mass spec software. Next slide, please. And this is how the schematic looks like. You have a similar design as I shown you for 4500. Only thing is that instead of a QJet, we have a new dual QJet technology, which enables a high focusing of all the ions which is getting generated in turbo V source. 45 and 55 both is coming with a turbo V source, which is one of the most important and most legendary ion source available in LC-MS market. And then you have a curved linear collision cell, and detector technology in this machine is also different, as I explained you. It is coming with HED detector, which is same as 6500 plus. So there is a lot of improvement in this 5500 plus we are having right now. I'll be giving you a little bit understanding about the Q-trap. You might have seen Q-trap mass spectrometer you may be using in your laboratories, but just for the larger audience, I just wanted to tell what the Q-trap means from the Sciex. So Q-trap stands for quadrupole plus ion trap, coming in a single mass spectrometer or in a single platform. And this is a hybrid mass spectrometer which Sciex invented a long time back and hold the patent for this technology. So basically this is a two mass analyzer sitting in one single mass spectrometer. So the importance of Q-trap mass spectrometer is that in various analytical R&D labs, different kind of samples comes. Sometimes samples for quantitation comes, sometimes samples for qualitative experiment comes. So as you know that trap is best technology for doing lot of qualitative experiment, while triple quadrupole is best for quantitation in MRM mode. So when you have both the mass analyzer sitting on one single platform, you can have a capability of running both type of experiment without compromising any data quality. So that is the USP of this particular Q-trap technology, that no matter what kind of sample is going to come in your lab, you are not going to lose any data quality, you are not going to compromise with any data quality, and in triple quadrupole or in Q-trap mode, you can able to run all the experiments. What you are seeing here in a small diagram, that you can scan in time manner, you can scan in a space manner, and when both things are combined together, you have a plethora of scans available. Scans from triple quadrupoles are available, scans from trap is available. So you have a long list of scans available which help you to perform and build any kind of experiment which mass spec is required. And in a small table what you are seeing, you are also able to see the difference between triple quadrupole and Q-trap scan. Q-trap holds multiple number of scan types, therefore it gives you flexibility to perform and build any type of experiment in your laboratory. Next slide please. And just this slide tells you that how isotopic patterns looks like on Q-trap mass spectrometer. This is the example showing for reserpine, where you can see a very nice isotopic pattern where peak width is around 0.15 AMU. So when you are running any characterization experiment, you can get this kind of isotopic pattern. Definitely this isotopic pattern is much better when you compare with a triple quadrupole, but definitely this is not like high resolution mass spectrometer, but when you talk on nominal mass platform, this is what you can this is what the best data you can get it from from Q-trap on ER mode. Next slide please. And even though not with the small molecules, if you are even running with a small protein peptide, a doubly, triply, and quadruply charged ions can also be resolved in a very appropriate manner using a Q-trap technology. And this is what the scans and isotopic resolution you are seeing on this current slide. And I don't want to go much more detail on nitrosamine, because already the examples and some of the things what Dr. Rao already has presented. Go to next slide. My introduction slide is a more or less same what Dr. Rao has presented. The introduction, you know that it's a basically nitroso functional group is bonded with any deprotonated amines, and the important thing is that nitrosamine is present in food and in drinking waters, published in many literature. But the problem comes when their presence is there in medicines, and which is not acceptable by any regulatory agencies. They say that presence of nitrosamine in any medicine should not be there at the moment. The potential impact on the business, what you are seeing initially, it is found in blood pressure medication, which is called angiotensin second receptor blocker, ARB, and then a certain one class of medicine like a sartan, various sartan product, it was detected there, and off lately then even ranitidine it was reported, and there is a worldwide review for various sartan and ranitidine, which led to the various recalls by the major pharma companies here in India and China. And that's why now all the companies are setting a very strict procedure to control the formation of these nitrosamine if it is there in their process. And also the LCMS is getting widely accepted, or especially triple quadrupole and Q-traps are widely accepted for doing the targeted quantitation or screening for these nitrosamine in various sartan and ranitidine products. And you can see the references I have listed down, and you can go and check. This is how the origin and chemical structure of nitrosamine looks like. This is the paper published in Journal Pharmaceutical or Biomedical Analysis in just 2019, where they say that how secondary amine react under acidic condition with nitrile and it forms. And what you are seeing in a small table, that various amines forms corresponding nitrosamines, and this is what very much available on the paper, on the various papers. You can go through and you can understand the chemistry part of it. And basically the important thing is the risk assessment is also very important for nitrosamine in various products. Next slide, please. And some of the regulatory numbers I have put it here for nitrosamine, as already Dr. Rao has mentioned about the specs level. These information came from US FDA. Initially, they were looking for 6 nitrosamine. The specs level was almost like 0.03 ppm, and then further some analytical levels was already defined by many laboratories like LOQ. They want to go to 0.01 ppm. LOD, they wanted to go around 0.005 ppm in all the sartan products, and then later even the NDMA came into ranitidine, and there the specs level was around 0.09, and subsequently the LOD and LOQ was also defined. And you can get more information about the limits from the US FDA website or from the EMA website. Go to next slide, please. And some of the LCMS method, what Sciex has already developed here for major sartan compounds, for Losartan, Losartan, candy sartan, and many sartans, but just because of the time constraint, I'm just able to show you the 3 data right away in this particular presentation. So this particular slide is for Losartan. Losartan, we have developed method on 4500 Q-trap and 5500 Q-trap, both machines. Here I'm showing you the data with 5500, where all the 6 compound was developed for the quantitative assay. And the method, what you are seeing is as per the regulatory limits, we can able to go up to 0.01 ppm LOD and 0.005 ppm of LOQ level. And the chromatographic condition is already being defined. You can see a very nice calibration curve of all the 6 compounds. Go to next slide, please. Similarly, if you see about Losartan we have done on 4500 with well-defined chromatographic conditions, and what you can see, we can finish this run in almost 19 minutes because you need a little bit time to flush the column for the second injection. All the peaks are very much separated from the Losartan API, and it's a very nice and very reproducible method what we have produced. And the limits, what you are seeing in the Sciex method is almost similar or better than what US FDA or European Union has published in their monographs. And this is a calibration curve where you are seeing a very nice statistical data at LOD level and also specification level. All is well within the range. Even accuracy, if you are seeing, is all within 75 to 110%. So it's all well within the range. Then last example is the ranitidine. Ranitidine also we have done with only 1 molecule, but even if somebody are interested to do more impurity in this, we can able to do always. But as per the regulation, we have just done with a 1 impurity, which is NDMA on 45 Q-trap. and it is a very well reproducible method. You can see the chromatographic condition and all the specification which is mentioned here. You can able to see that we have used 2 transition for NDMA to understand how the ion ratio looks like, how the consistency looks like in a longer run. And yeah, this is the chromatogram. This is the chromatogram. You can see the calibration curve, and you can also able to see the statistical data at LOD, LOQ, and specs level. It's all within the range. The precision and accuracy batches are all within the defined criteria by the regulators. And Q-trap workflow, I just wanted to take a couple of minutes to explain what the Q-trap is. As I told you already, that Q-trap is not like a triple quadrupole. It's a hybrid mass spectrometer where quadrupole and trap, which is basically a 2 mass analyzer sitting on single mass spectrometer. So you can use both mass analyzer as and when it's required in your laboratory to perform qualitative and quantitative application. We have run some of the screening experiment to understand that how we can quickly screen the nitrosamine in the various batches using a Q-trap. So next slide, please. And what you are seeing here is that you can, we have IDA workflow, which is called information dependent acquisition workflow. This workflow help you to on-fly acquire 4 set of data like full scan spectra, isotopic pattern, product ion, and MS, and this all can acquire in a single injection, and that's the beauty of this particular workflow. What you are seeing on extreme right, that we are acquiring MRM data, we are acquiring ER data, and we are acquiring enhanced product ion spectra in a single run. And then what you will see in the next slide, that enhanced product ion scans, we are using that information from the library to match and give you the information about the impurity which is present based on the matching score from the library. So this can help you to quickly screen the compound in non-targeted way, just to have understanding based on the information available on the library and the data what you are acquiring. So the Q-trap help to do all these kind of non-targeted screening as well in your day-to-day samples. Next slide, please. And some of the global reference LCMS method, there are various regulators throughout the globe, like US FDA, European agencies, Canada Health, Singapore agencies. They already publish so many reference paper. You can go and Google them. But some of the papers, what we have listed here is from EDQM. EDQM used a 5500 Q-trap mass spectrometer, and they have developed a lot of method on these sartans, a very sensitive, very rugged method they have published when this all fiasco started. Next slide, please. And this is the reference. You can go and you can understand, you can download all the methods from EDQM. And very recently, you might have seen this particular tech note, or we can share with you. You can write to marketing team. This is the paper or the full blown validated method was published by Sciex application support team on nitrosamine in various sartan, and even ranitidine. So my request to you is that just Google this one, or just write to the marketing.india@sciex.com and get your copies. And if you need any support developing this method, or you need any support from our app support team, we will be happy to assist you. And last not least, but this presentation would not be possible without the support from my application support team. The name listed here, you can see from various people supports are required to make generate this data. So Dr. Manoj Pillai, Aman, Sandeep, Chandrasekhar, Pankaj, and my very enthusiastic marketing team, Parikshit and Valerie. Thank you guys for helping and bringing this webinar. And then we have our EMEA team, Ferran, Gianru, and Jack. they all helped us to put all this slide deck together. With this, I'm end on my presentation. Probably we can have some question now.
Yeah, no?